Hello everyone and welcome back to our last episode about photosynthesis. Previously we talked about the light-dependent reactions of photosynthesis along with the Calvin cycle, and now we're talking about adaptations to photosynthesis. So in the last video we ended off talking about how RuBisCO, that enzyme that's really important in the Calvin cycle, binds to oxygen as well as carbon dioxide. And that process, called photorespiration, wastes all the products of the light-dependent reactions. And there are ways that we can remedy that, but some plants have evolved to reduce the rate of photorespiration that occurs to begin with. So in really hot environments, plants are going to be closing their stomata to conserve water, and that results in a buildup of oxygen, and they don't get enough CO2. So with these really high oxygen, low CO2 environments, we're going to see a lot of photorespiration occurring, and plants have evolved to prevent that. So the question is, how do we prevent RuBisCO from binding with oxygen? And there's a few ways we can do this, but both of them use this really cool enzyme called PEP carboxylase. Okay, so both of them use PEP carboxylase, which is an enzyme that binds things to carbon. So RuBisCO, this guy is both a carboxylase and an oxygenase, meaning that it binds both carbon molecules and oxygen molecules. When it binds with carbon dioxide, that's the carboxylase activity, but when it binds with oxygen, that's the oxygenase activity. And that's bad. That's the photorespiration we don't want to happen. So instead we can utilize a carboxylase, which only binds with carbon molecules. So both of our methods are going to harness this guy named PEP carboxylase. Okay, so we're going to draw two different cells here, because what C4 plants do is they separate different reactions into different cells. And you'll see why that's helpful. Okay, so this cell here is a mesophyll cell. And this is a bundle sheath cell. You don't really need to know everything about mesophyll and bundle sheath cells, most likely, unless you're in botany, but it is important to know that these are happening in different cells. So we start with CO2, that part is the same, and that's coming from like our stomata being open. We collect some CO2, and it gets bound to this carbon molecule called PEP. It's a 3-carbon molecule, just like RuBip is, and it gets bound with carbon dioxide to form oxaloacetate. And guess who is binding it? Oh, is oxaloacetate gonna fit in here? It's gonna go outside, that's okay. The enzyme that does this is PEP carboxylase. I'm gonna say Pep C. That's just the thing that's allowing this to happen. This reaction is catalyzed by PEP carboxylase. Okay, so now we've made oxaloacetate, and so this is 1 carbon, this is 3 carbons, so oxaloacetate is 4 carbons, which is how we get the name C4, is the carbon molecules we're working with have 4 carbons rather than the normal C3 pathway, which is obviously a 3-carbon molecule. Okay, and then that gets turned into malate, which is another 4-carbon molecule, and that will continue on to this other cell. So malate will be delivered to the bundle sheath cell. So malate will be turned into carbon dioxide and another 3-carbon molecule called pyruvate. And pyruvate is turned into PEP, which is how we started from the beginning. Okay, so through this process, we started with CO2 and we ended with CO2. So what's the point of that? Well, since PEP carboxylase only binds with carbon dioxide, we can have a ton of oxygen in here and PEP carboxylase won't even care. So we can have a bunch of oxygen in here and it won't be a problem. PEP carboxylase will ignore it. And it'll only take the CO2, and then we can put it into here and only put the CO2 in here. And then we can go through the Calvin cycle, and RuBisCO can function normally without having to worry about oxygen because there is no oxygen. So we do the Calvin cycle, we make glucose, and there's no photorespiration occurring. Since oxygen is never introduced to this cell, it's not a problem, and that's all thanks to PEP carboxylase. So it seems like a lot of steps just to start with CO2 and end with CO2, but what it's basically doing is filtering out any oxygen so that RuBisCO isn't affected by it. So that's the point of the C4 pathway, is we separate oxygen from the Calvin cycle to prevent photorespiration. And also what's kind of cool is the light-dependent reactions occur a lot in the mesophyll cells, and that produces oxygen. So this way, we're also separating the oxygen we get from the light-dependent reactions from the Calvin cycle with RuBisCO. Okay. Oh, maybe I should highlight that with the other green. Hang on. I was just highlighting my terms, but okay. Mesophyll cell, bundle sheath cell. So what's really important here is our PEP carboxylase turning our PEP into oxaloacetate. And these 4 carbon molecules is why we see C4 in this pathway. So with this spatial solution, separating it in space, the oxygen from the RuBisCO, we see less photorespiration occurring. However, all these other reactions do take some energy. So since we're doing all these reactions just to end up with the CO2 we started with, it is a little bit less efficient. We end up using 5 ATP to fixate 1 CO2. I'll say per CO2 fixation. And the normal C3 pathway uses 3. But this, although it's still a loss compared to the normal pathway, but in a hot environment where we have to close our stomata, we don't let in any CO2 and we're not letting out any oxygen, it's a lot more efficient because we're preventing photorespiration from occurring. Okay, so what is our other method? These are called CAM plants, which are actually really easy to remember because the solution with CAM plants is you separate it into day and night. So I like to remember CAM has AM, aka like AM, PM, so night and day. So this is a spatial solution, and this is a temporal solution based off of the time of the day. Okay, so what does that look like? Okay, we're gonna draw a big mesophyll cell, which we can use this label from over here because it's still a mesophyll cell. And similar to the C4 pathway, we start with PEP, that 3-carbon molecule, and using PEP carboxylase, we can say PEP C. We let in some carbon dioxide. So at night, the plant stomata will be open because it's night, it's not as hot, we're not going to be losing so much water. So these are supposed to be our stomata. There's like an open pore here, right? So carbon dioxide is coming from the environment, it's entering the cell, and that's okay because it's night. So right now we're looking at the reactions that happen at night. Okay, so PEP carboxylase is going to bind PEP and carbon dioxide, same thing as what's happening here, and that's going to once again form oxaloacetate. And just like before, it's also going to turn into malate. So the malate is actually going to be converted into this thing called malic acid. And that's going to be stored in a vacuole. We can color this vacuole in. And remember, this is all happening at night. This is a vacuole, don't forget. Okay. So this is the kind of thing we see happening at night. Since it's not too hot, the stomata are open, and then the carbon dioxide can come in, and we can do this reaction and store some malic acid. And then during the day, we can use that malic acid, turn it back into malate, and make sugar, which is the whole point of photosynthesis, is making energy from the sunlight. So if you want, you can kind of distinguish what steps are where. So the day is happening here as we take the malic acid from the vacuole, turn it into malate, and turn it into sugar. So in the day, we can close our stomata. We don't need the CO2 because we have this built up malic acid to make sugar out of. But at night, we need to open it up to get the CO2 in, let any oxygen out, and build up that malic acid. So this is another great solution. You can see it mostly in very wet, like succulent-y plants. So like watery, succulent plants, so like cacti. And if you want to see an example of a C4 plant, that'll be something like maize or corn. And if you've ever heard of sorghum and teff, those are some C4 plants. So you'll see this in a nice sunny environment, but definitely the cam plants are in a very like deserty environment like cacti. They're going to store a lot of water. That's why their stomata are closed all day, is because they need to store that water or else they'll dry up. And since they can only do one thing at a time, they're pretty slow growing, which we see in various forms of cacti. Also, it's interesting to know, with both of these adaptations, it can kind of slow the plants down. As you know, this C4 pathway uses more ATP than normal carbon fixation, but we know that it works because it's evolved more than 19 times. Meanwhile, the cam plants have evolved over 23 times. Sorry, I'm a little inconsistent there. You can use the greater than symbol or over. I guess I decided to use both. But anyway, so these are our adaptations to photosynthesis. The C4 pathway is a spatial solution. The CAM pathway is the temporal solution. Oh, and if you want to know what CAM stands for, we can write that down. So CAM stands for crassulation acid metabolism. This word crassulation, it's referring to Crassulaceae, which is a family of succulents that are CAM plants. The acid is referring to the acid buildup that happens all night of that malic acid. That's a way to remember it, but I've never been in a class where you actually had to know what CAM stands for, but that is kind of a cool way to remember CAM plants from C4. So remember, C4 is called C4 because we have our 4 carbon molecules that we're using to get CO2 to bind with the Calvin cycle. Meanwhile, CAM, you can remember from AM, like AM versus PM, because it's a day versus night thing, so AM/PM. But also you have Crassulaceae, which is that family of succulents that do CAM photosynthesis, and they have metabolism of this malic acid. All right, so yes, we know that it works because it's evolved so many times, lots and lots of times, because in really hot environments, you can't afford to lose all your products in photorespiration. Some examples are corn, sorghum, and teff. You'll see corn the most often, whereas you'll see like cacti and succulents more often for CAM plants. So yeah, this answers our question, how to prevent RuBisCO from binding with oxygen, our carboxylase plus oxygenase. The answer is to use a carboxylase instead. Well, you still use RuBisCO in the Calvin cycle. So these are pretty simplified diagrams. I guess we could say like the Calvin cycle happens here too. Calvin cycle. Because it's the same thing that happens in these C4 plants, is we take malate, put it through the Calvin cycle, and make sugar. So here I say glucose, here I say sugar, but they're really the same thing. You could write glucose instead because glucose is a type of sugar. But yeah, that's the idea. Those are our solutions to photorespiration, our adaptations to photosynthesis, and our last lecture on photosynthesis. But fear not, there's a playlist full of other videos on biology you can watch, some made by me, some not, but they're all the same idea. Yeah, so thank you for watching, and I will see you later.